Hi there. Our conscious awareness of incoming sensory information is called sensation. Only a stimulus that reaches the cerebral cortex of the brain results in a sensation of that stimulus. Thus, although our bodies are constantly bombarded by sensory stimuli, we are consciously aware of only a fraction of these stimuli. Stimuli are detected by receptors. This chapter focuses on the general senses receptors: temperature, pain, touch, stretch or vibration, and pressure. So what are receptors? Recipio equals to receive. Receptors are structures that detect stimuli. They range in complexity from the relatively simple dendritic ending of a sensory neuron to complex structures called sense organs, whose nerve endings are associated with epithelium, connective tissue, or muscular tissue. Receptors in the body monitor both external and internal environment conditions and conduct information about those stimuli to the central nervous system. We are aware of some specific stimuli. Other stimuli never reach our consciousness. The receptive field of a receptor in the entire area through which the sensitive ends of the receptor cell are distributed. There is an inverse relationship between the size of the receptive field and our ability to identify the exact location of a stimulus. If the receptive field is small, precise location and sensitivity are easily determined. In contrast, a broad receptive field only detects the general region of the stimulus. There are several types of receptors. Chemoreceptors detect chemicals such as specific molecules dissolved in fluid in our external and internal environments, including ingested food and drink, body fluids, and inhaled air. For example, the receptors in the taste buds on our tongue are chemoreceptors because they respond to the specific molecules in ingested food. Likewise, chemoreceptors in some of our blood vessels monitor the concentration of oxygen and carbon dioxide molecules in our blood. Next type are thermoreceptors. They respond to changes in temperature. Photoreceptors are located in the eye, where they detect changes in light intensity, color, and movement. Next type are mechanoreceptors. They respond to touch, pressure, vibration, and the stretch. Most of the cutaneous receptors are mechanoreceptors because they respond to pressure and touch on the skin. Mechanoreceptors are also located in the ear for equilibrium and hearing. Baroreceptors detect changes in pressure within body structures. These sensory receptors branch repeatedly within the connective tissues in vessel or organ walls, especially the elastic layers. Any pressure stimulus that causes wall deformation results in a change in the nerve impulse rates from the receptors and the pressure sensation. Nociceptors or nocireceptors respond to pain caused by either external or internal stimuli. Somatic nociceptors detect chemical, heat, or mechanical damage to the body surface or skeletal muscles. For example, exposure to acid, touching a hot pan, or suffering sprained ankle stimulates somatic nociceptors. Visceral nociceptors detect internal body damage within the viscera due to excessive stretching of smooth muscle, oxygen deprivation of the tissue, or chemical released from damaged tissue. Clinically significant types of pain. Phantom pain is a sensation associated with a body part that has been removed. Following the amputation of an appendage, the patient often continues to experience pain that feels like it's coming from the removed part. The stimulation of a sensory neuron pathway from the removed limb anywhere on the remaining intact portion of the pathway propagates nerve impulses and conducts them to the CNS, where they are interpreted as originating in the amputated limb. In other words, the cell bodies of the sensory neurons that provides sensation to the leg remain alive because they were not part of the leg. This so-called phantom limb syndrome can be quite debilitating. Referred pain occurs when impulses from certain viscera and perceived as originating not from the organ but in dermatomes. Numerous cutaneous and visceral sensory neurons conduct nerve impulses through the same ascending tracts within the spinal cord. As a result, impulses conducted along ascending pathways may be localized incorrectly. Consequently, the sensory cortex in the brain is unable to differentiate between the actual and false sources of the stimulus. Clinically, some common sites of referred pain are useful in medical diagnosis. For example, cardiac problems are often a source of referred pain because the heart receives its sympathetic innervation from the T1 till T5 segments of the spinal cord. Visceral pain is usually referred along the sympathetic nerve pathways, but sometimes it can follow parasympathetic pathways as well. For example, referred pain from the urinary bladder can follow the parasympathetic pathway via the pelvic splanchnic nerves. Since the pelvic splanchnic nerves lie in the S2 till S4 segments of the spinal cord, pain may be referred to the S2, S4 dermatomes which overlie the medial buttocks region. Tactile receptors. They are mechanoreceptors that react to touch, pressure, and vibration stimuli. They are located in the dermis and the subcutaneous layer. Tactile receptors range from simple dendritic ends that have no connective tissue wrapping called unencapsulated to complex structures that are wrapped with connective tissue or glial cells called encapsulated. Unencapsulated receptors have no connective tissue wrapping around them and are relatively simple in structure. The three types of unencapsulated receptors are free nerve endings, root hair plexuses, and tactile discs. Free nerve endings are terminal branches of dendrites. They are the least complex of the tactile receptors and reside closest to the surface of the skin, usually in the papillary layer of the dermis. Often, some dendritic branches extend into the deepest epidermal strata and terminate between the epithelial cells. These tactile receptors primarily detect pain and temperature stimuli, but some also detect light touch and pressure. Root hair plexuses are specialized free nerve endings that form a web-like sheath around hair follicles in the reticular layer on the dermis. Any movement or displacement on the hair changes the arrangement of these branching dendrites, initiating a nerve impulse. These receptors quickly adapt. Thus, although we feel the initial contact of a long-sleeved shirt on our arm hairs when we put on the garment, our conscious awareness subsides immediately until we move and the root hair plexuses are restimulated. Tactile discs, previously called Merkel discs, are flattened nerve endings that function as tonic receptors for fine touch. These receptors are important in distinguishing the texture and shape of a stimulating agent. Tactile discs are associated with special tactile cells, Merkel cells, which are located in the stratum basale of the epidermis. Tactile cells exhibit a small receptive field and communicate directly with the dendrites of a sensory neuron. Encapsulated tactile receptors are covered either by connective tissue or glial cells. Encapsulated tactile receptors include Krause bulbs, lamellated corpuscles, Ruffini corpuscles, and tactile corpuscles. Krause bulbs are located near the border of the stratified squamous epithelium in the mucous membranes of the oral cavity, nasal cavity, vagina, and anal canal, where they detect light pressure stimuli and low frequency vibration. Lamellated corpuscles, previously called Pacinium corpuscles, are large receptors that detect deep pressure and high frequency vibration. The center of the receptor houses several dendritic endings of sensory neurons within numerous concentric layers of flat fibroblast-like cells. This structure ensures that only deep pressure stimuli will activate the receptor. Lamellated corpuscles are found deep within the reticular layer of the dermis. In the subcutaneous layer of the palms of the hands, soles of the feet, breasts, and external genitalia, in the synovial membranes of joints, and in the walls of some organs. Ruffini corpuscles detect both continuous deep pressure and distortion in the skin. There are tonic receptors that do not exhibit adaptation. They are housed within the dermis and subcutaneous layer. Tactile corpuscles, previously called Meissner corpuscles, are physically different from the unencapsulated tactile discs discussed previously. Tactile receptors are large and encapsulated. Oval receptors, next type of receptors, formed from highly intertwined dendrites enclosed by modified neurolimocytes, which are then covered with dense, irregular connective tissue. Tactile corpuscles are phasic receptors for light, touch, shapes, and texture. They are housed within the dermal papillae of the skin, especially in the lips, palms, eyelids, nipples, and genitals. That's it, dear students, about receptors of general senses. Good luck, see you.